So far, we've focused on the retina in terms of converting a photon of light into first a receptor potential in the photoreceptor, and then how the photoreceptor, via communication with the bipolar cell and the retinal ganglion cell, conveys additional information. Each step along the pathway, more and more information is added in so that our ability to perceive photons of light gets more and more complicated and closer to the way we actually perceive the world. As we said before, information from the right visual field ends up in the left visual cortex. In order to get there, some information, that from the nasal retina, has to cross over at the optic chiasm. It first goes to the lateral geniculate nucleus of the thalamus. This is the sensory relay nucleus for vision. Information can then go to the primary visual cortex in the telencephalon. Although it's not shown in this particular figure, information from the retina also goes to the hypothalamus, in particular the suprachiasmatic nucleus of the hypothalamus. This nucleus is not important in the perception of visual information, but in circadian rhythm. Information from the retina also goes to the superior colliculus in the tectum of the midbrain in the mesencephalon. Again, it's not shown here because it is ventral to this particular horizontal section of the brain. The superior colliculus is important in visual reflexes. All of these pieces of information get sent to the primary visual cortex for additional processing. The way information is organized is still based on how it was organized in the retina. The parvocellular neurons, the parvocellular retinal ganglion cells, continue to be tracked together. Remember, this is the cone pathway. Each of these cells has a relatively small receptive field, and because each cell has a small receptive field, we need more cells to cover the entirety of the visual field. That's why when we look in the lateral geniculate nucleus of the thalamus, we have 4 layers of cells devoted to the parvocellular pathway. The magnocellular retinal ganglion cells are bigger cells. There's more convergence in that pathway, so we need fewer cells to account for the entirety of the visual field. Therefore, we only have 2 layers in the lateral geniculate nucleus of the thalamus devoted to those magnocellular inputs. We have another layer here as well called coniocellular. Coniocellular also is information from the parvocellular retinal ganglion cells, and it's sort of in between these parvocellular layers and in between the magnocellular layers. The coniocellular layers are important particularly in color sensitivity and in shape and form discrimination. Once we get to the primary visual cortex, we are looking at the calcarine fissure. That's why they're showing you a horizontal view of the brain here so that you can see this calcarine fissure. From the outside of the cortex, you can't see much of the primary visual cortex at all. Remember that the primary visual cortex is also sometimes called the striate cortex, or also called V1. This is a figure from Neuroscience Online showing the organization of the primary visual cortex. On the retina at the back of the eye, we essentially have a map of the visual field. That map is maintained all the way up to V1, to the primary visual cortex. So if this is the visual field, we can see how information from the visual field is mapped onto the calcarine fissure. If you have damage to the primary visual cortex, you will not have any conscious awareness of an ability to see. But you might still have some visual reflexes because those don't require conscious awareness. This is a table from Carlson listing the properties of the magnocellular, parvocellular, and coniocellular divisions of the visual system. Remember that parvocellular and coniocellular are both part of the cone pathway. In terms of sensitivity to contrast, remember that cones require high levels of illumination in order to respond to light at all. That means that cones are not good at differentiating different levels of brightness. And that holds true for the cone pathway. Rods, on the other hand, can determine differences in brightness from very low to higher. That's why the magnocellular division, which gets its information from rods, has high sensitivity to contrast. It can distinguish between low, medium, and high brightness. Spatial resolution refers to spatial acuity. Again, because it gets its information from the rod pathway, the magnocellular division has low spatial resolution. It has low acuity, whereas the parvocellular division has high acuity. The coniocellular division here is listed as having low acuity, even though it gets information from cones. We aren't going to talk a whole lot more about the conicellular pathway. As far as temporal resolution, this refers to how quickly the cells adapt to what they're seeing. The parvocellular and coniocellular divisions have slow, sustained responses. This is most evident in that example where you look at the white plus on the apple that is initially on the page shown as a green apple with a reddish purple leaf. Because the cells in these pathways slowly respond, when you move your gaze to the black plus on the other side of the page, you see the inverse image. In terms of the magnocellular pathway, the temporal resolution being fast means that the cells in this pathway will quickly respond to the onset and offset of light. That means that this pathway is better at providing information about movement of light. Whereas in this pathway, the cells will have a slow reaction. Even as the light moves across the pathway, there might still be a sustained response because they cannot quickly change their activity based on the presence and absence of light, the way the magnocellular pathway can. This is relevant also as we go into the visual association areas. There are two streams of analyses in the visual association system. The first is called the dorsal stream. Information goes from the primary visual cortex and travels dorsal. Most of the information in this pathway comes from the magnocellular pathways. It conveys information about light-dark contrast and movement, because those are the pieces of information that the magnocellular pathway understands. The ventral stream travels from the calcarean fissure, from the primary visual cortex, to ventral portions of the cortex. There's a big problem with this figure in Carlson, and that is that the arrow appears to go to the pons. The ventral stream does not go to the pons. It goes to parts of the cortex that are lateral to the pons. They should have ghosted in the cortex and shown you more where that actually goes. The ventral stream receives information from all of the pathways, red, green, blue in the parvocellular and conicellular systems, and also information about light-dark contrast and movement from the magnocellular division. The ventral stream is most important in object identification, whereas the dorsal stream is most important in telling you where an object is. We'll see in the auditory system how these pathways line up with the auditory association pathways. This is a better image of the ventral stream. Here you can clearly see that information is coming from V1 and traveling ventro into the inferotemporal cortex, not to the pons. Because this pathway is important in object identification, if you have damage to this pathway, you have a problem called aperceptive visual agnosia, inability to identify objects by sight. So if you looked at that picture of the man made out of fruits and vegetables, you could see different colors and shapes, but you would not be able to identify the fruits, vegetables, and flowers. This is a figure from Carlson showing a particular case study of somebody with damage to parts of what's called the extrastriate cortex. That's another way of saying the visual association cortex. And the particular symptoms experienced are loss of form but not color. Again, they can identify colors and maybe shapes, but can't identify specific objects. As you go farther and farther along in the ventral stream, the information that you are processing gets more and more detailed. At the very end of the ventral stream is a brain region called the fusiform face area. This is a view of the brain as if you're looking up at it. This is where the brain stem would connect to the spinal cord. So we're at the ventralmost part of the cortex. The fusiform face area, just like its name sounds, is activated by familiar visual stimuli relevant to faces. Our brains are primed to identify faces because we are a social species. That's why it looks like there's a man on the moon, or we see a face in the clouds. If you have damage to the fusiform face area in the ventral visual stream, you have a particular disorder called prosopagnosia, inability to recognize faces. There's a great case study in the Oliver Sacks book, Man Who Mistook His Wife for a Hat, where he describes a man who literally mistakes his wife for a hat. He doesn't recognize her face. He sees something next to him and thinks it's his hat. If you have prosopagnosia, that's not the same as aperceptive visual agnosia. So if you have prosopagnosia, you would still see a pear and a flower and whatever other fruits and vegetables these are made out of, but you would not see a face. The dorsal stream in the visual association cortex is most important for motion perception. And there are studies with humans showing that if you have damage to this part of the brain, you can have a problem called akinetopsia, inability to perceive movement. And that's caused by damage to area V5, which is also sometimes called MST.